27 and chapter 38.
What?
27 and chapter 38. We've already done a lot of the chapters in the 20s earlier in the semester.
I remember we did animal behavior, it was like something.
Animal behavior was like in the 40s, I think. But then we did, I think, chapters 24, 25, and 26 earlier in the semester, maybe in November.
To be honest, I don't remember what the chapter numbers are. I just remember.
I don't either. I don't blame you. Okay. Chapter 27 is called Bacteria and Archaea. And the only portion of chapter 27 that we're going to talk about is just prokaryotes in general and bacteria specifically. Where we're not going to really talk about Archaea because it very rarely, if ever, comes up on the AP exam, and when it does, it's like just data. It's you know, you're looking at a figure or a table, and you don't really need to know anything about Archaea. All right, but there are a few things we need to know about bacteria. And first question that we're going to address from chapter 27 is, where are they? Where do we find bacteria? Where do we find bacteria? Where do we find archaeans? Where do we find prokaryotes in general? Where do we find these organisms? And where are some of the places we find bacteria? In our bodies. Love it. So up until, I don't know, maybe about 5 years ago, it was thought that you had more bacterial cells in your body than you had human cells. We now know that it's not quite the case, but it's pretty close. You have literally trillions of your own cells in your body, and bless you, and you have trillions of bacterial cells in your body, lining every surface. You've got bacteria in your gut, you have bacteria in your ear canals, you've got bacteria in your nose, you've got bacteria in your mouth, you've got bacteria lining cavities inside. I mean, you have bacteria all over the surface of your skin. It's wonderful. You got bacteria in your eye, bacteria in our hair. Where else do we find bacteria? Food. And this is probably the, are you thinking in terms of creating issues? Like if it's contaminated, like you get E. coli poisoning from eating contaminated lettuce from Chipotle?
Normal bacteria that's like not harmful.
Okay, yeah.
Chipotle.
Chipotle. The I is pronounced as an E because it's Spanish. Okay, so the only real connection, I mean, there are two connections between chapters 28 and 27 and 38, and that's bacteria and flowering plants are both living. Cool. The only other connection is that they are both incredibly important in agriculture. Incredibly important. So we use bacteria to make food. Cheese, wine, bread. Blue cheese is a fungus. What's that? Blue cheese? It's a little tart. It's a little tart. I like it. I like it a lot. Okay, and so we definitely find bacteria in our food, both good bacteria and bad bacteria. Yogurt, you can't make yogurt without bacteria. I mean, I could live without yogurt, but some could. You have another answer?
Cottage cheese is gross.
Okay, relax. Mark, where's another place we find bacteria? What's that? Water. Thank you. Okay. We find bacteria in water, and we find archaeans in water, and in some waters, bacteria and archaeans are the only living things you find. In waters that are really, really hot, and in waters that are really, really cold, pretty much the only living material you find are bacteria and archaea. Okay? In water that's really, really salty, it's pretty much the only thing you find, bacteria and Archaea. Yep. In water that's really, really acidic, it's pretty much the only thing that you find. Okay? And since we're getting another big place where we find lots and lots of bacteria in the soil. And so typically, if you want to have a really strong, healthy garden, that's one of the things you monitor, is you check your soil and you monitor it for the amount of bacteria present in the soil. Both good bacteria and bad bacteria. Yeah, Trinity.
Is there more good than bad?
In terms of number of species, yes. Or I would say there's more, if you lump good and we don't know together, there's way more of those than we're like, oh, these are definitely bad. These are pathogenic.
Is there more than like you knows than you don't know?
No. Yeah, there are several species of bacteria that we don't know what they look like. We've never grown them in the lab, but we found their DNA in soil somewhere. Yeah. Emma.
So like if you know what they look like, is it easy to tell if they're good or bad?
Sometimes, yeah. I mean, there are some features, and we'll talk about that in a minute. Not literally a minute, but in a period of time. There are some things that you can look at that are pretty good clues. Yeah, pretty good clues. All right. So where are they? Yeah, Michelle. Air. A little bit, but not a ton. So they can get suspended up in the air, but they don't really live there. And so the answer to where are they is basically everywhere. If you were to eliminate all living organisms except bacteria and Archaea, those were the only organisms you kept, only prokaryotes, you would still find every environment on Earth filled with life. And you'd find the soil all over the Earth, the water all over the Earth, including great depths, and it would still be full of life. Now it would be microscopic life, and you wouldn't see it, but you wouldn't be there because all living things are gone except for them anyways. Yeah. Alright, this is a hypothetical. It's a pretty simple question to answer. Where are they? Everywhere. And interacting in ways that we aren't even fully aware of yet. Okay? Any questions about this question? It's a pretty simple question, pretty straightforward question. The next one's a little bit more complicated. Next one's a little bit more complicated. Why are they so successful? Why do we find them in places where we don't find any other living organisms? Why do we find them in waters that are at 90 degrees Celsius where nothing else can live? Why do we find them at waters that are at 4 degrees Celsius where nothing else can live? Why do we find them in waters that have a pH of 3, which is pretty acidic, where nothing else can live? Why do we find them in waters where they're so salty, salt's literally falling out of solution? And they don't care. I mean, they love it. They just absolutely love it. Michelle.
Because they can't easily adapt to it.
Yes, they adapt very quickly. Is that what you're
I
gonna?
was gonna say, like, the things that they need to survive is really simple. Like, they don't need much.
Oh yeah, I mean that's a really good point as well. And so they adapt quickly. It's an excellent point to make, and part of that is because they're simpler, right? They don't need everything we need in order to survive. And then part of that is just because of 3 features. One is they have a rapid reproductive rate. We'll say rapid reproduction. Bacteria reproduce by binary fission. Binary meaning what? 2, and fission to split or to break. And so you take one cell, you replicate the genome, and you split it into 2. Okay? And it takes E. coli, which is the bacteria we know best, because it's a bacteria we live with all over our bodies. Thank you, Chipotle. It takes E. coli 20 minutes to double. Every 20 minutes, the cell number doubles.
Oh my god.
So if you took one
Good day.
cell of E. coli, in a day you'd have hundreds of millions. Okay? Very quickly, rapid reproduction.
Is it
How is the whole world just not equal?
Oh, so it's exponential.
Viruses.
How is the
Exponential growth.
Viruses.
Because there are viruses that hold bacteria in check. There are things that eat bacteria.
Like if like bacteria were the first order, the viruses would be the resistance.
So
Okay.
1. Yeah, 2. No, 16.
It's important.
Another reason they are less resistant to mutations, and that's not saying that they just love to mutate, but they're less resistant to it. So eukaryotes, when you get a mutation in a eukaryotic genome, it's very easily devastating. You get a mutation in a prokaryotic genome, they can usually handle it a little bit better. Plus, on top of that, they're reproducing so quickly that mutations can accumulate relatively fast. Relatively fast. And then the third thing is genetic recombination is common. And somebody remind me what genetic recombination is. We've seen it once in this class. Katie.
Is it where you take parts from each other's genomes?
Yes, parts from your mom's chromosome 1, parts from your dad's chromosome 1, and you recombine them into a chromosome 1 that never existed before.
Yeah.
Right? It's like if you had 2 decks of cards, and they're a little bit different, you know? They're a little bit different, and then you mix them together, but to still only get 52 cards. But you've got cards from 2 different decks. Cards from 2 different decks. And so genetic recombination is really common. Now, it's common in our gametes to find examples of genetic recombination, but it's very rare for, let's say, you to take my genome and during my life I get genes from some other human being. It's very rare. The only way for it to really happen is for, say, Emma to be infected by a virus. That virus moves to me and brings some of Emma with it. Why did I use you as an example? I don't know. Is that what you were wondering?
I said maybe it'll make me stop talking.
It would make me talk more. Yeah. It would make me talk more. Okay, and so genetic recombination is really common in bacteria. We've already seen this, right? We saw our R-type bacteria when they are exposed to dead S-type bacteria. What happened to those R-type? They become like the S-type. Okay, genetic recombination. They took some genetic material from dead S-type bacteria and they became S-type bacteria, right? We also saw it with the Hershey-Chase experiment where you use a virus to transduce a bacterium and make a genetic recombinant. Right? Right?
Right.
What type of bacteria, or no, sorry. Not bacteria. What types of viruses insert their genetic material into the host?
Oh my gosh, so
Lysogenic, right? Lytic viruses, they convert the host into a virus-making machine and blow them up. Lysogenic viruses put their genome into the host genome. Okay? So it's another way in which bacteria can go through genetic recombination. And I'll show you this in just a little bit. Yeah, Michelle.
How is it helpful to be like successful?
How is it helpful to be successful? Well, it could be harmful, right, when you get new genetic material, or it could be helpful. What it means is it facilitates getting combinations that are new and unique and may be able to survive in different kinds of environments.
Also, they have like different traits.
Yes.
Okay.
Yep, absolutely. All right. We're going to come back to these, and I'm going to show you 2 of our 3 mechanisms for genetic recombination. Transformation is pretty simple, right? What is transformation? This is what the Griffith experiment showed, where our R-type bacteria became S-type. Look what's happening there. How did they become S-type? They drank up some of that S-type DNA and then incorporated it. I'll show you the other 2. I'll show you pictures of them and you'll love it. You'll love it. Okay, any questions about this question? Making good time. No? Alright.
We have a random question.
Who has a random question? Oh, yeah, what's up?
So at lunch yesterday, someone said that if you eat oranges, it like prevents cancer, like or orange peels or something. Is that because the vitamins?
on their skin, you think.
Yeah.
Yeah, you know, so what, first of all, I would say, I would respond if I heard somebody saying that to, I want to see the data. That would be my first response. I want to see the data where they've actually demonstrated a link between consuming oranges and prevalence of cancer. So I'd want to see the data. Secondly, even if I saw the data, I would be cautious because the only way to make a kind of claim, if somebody said, first of all, if somebody said eating oranges prevents cancer, I would respond by saying, one, I want to see the data, and two, you can't make that kind of a claim statistically.
Yeah.
You could say there's a correlation between eating oranges and lower rates of cancer. But yeah, the only way to say that eating oranges prevents cancer is to do a controlled experiment.
And you can't do that.
Well, I mean, so eating oranges, there's nothing like really concerning about that, right? But then the problem is that it'd be very difficult to give somebody a placebo where they think they're eating oranges, but they're not really eating oranges, right? So it's like, I mean, what do you give them? It's like, wait, we're gonna blindfold you, and we're gonna give you something that's like a gummy. One of them actually has real orange essence, and the other one has like artificial orange essence. I mean, it's just gonna be really challenging. And then so it's like, well, are these people having lower rates of cancer because they really believe that they're healthier?
Yeah.
And when people believe that they're healthier, they tend to have a more healthy lifestyle. And then so it's just like, I don't know. So I'd want to see the data, and two, I would be very cautious to accept that. And then I would consider the source, thirdly. And so if it's like something.
But if that did work, would it be because the vitamins?
So full refutation.
So if you actually were able to demonstrate that there's a very significant correlation between consuming oranges and lower rates of cancer, I would say if you could actually attribute it truly where you were pretty confident it was because of the oranges, it would probably be something in there that is acting as an antioxidant that is limiting the mutation rate. Emma, is this another random question?
It's not like
What else
cancer.
would it
Oh, okay.
And things like
That's
that.
not really a random question.
It's kind of stupid, but like, how can they not find cures for things? Can't they just look at like the makeup of something and then see like what? Yeah. Like.
Well, it's pretty complicated. So let's say something like a pathogen. Since we're talking about bacteria, let's say something like a pathogen. Like you've got a particular bacterial illness that's very difficult to treat. Let's say methicillin-resistant Staphylococcus aureus.
I was gonna say staph.
MRSA.
Oh, that's it. Why did you say
I have a question. Actually, can I ask another question?
So you don't need to write this down. I'm just putting this here. Although we will actually use this as an example of rapid evolution rate and an outcome of it. So methicillin-resistant Staph aureus. They cause a staph infection, but they're resistant to antibiotics. And so what the problem here is because these bacteria replicate so quickly, it's very difficult to eradicate them fast. Even if you have a really effective antibiotic, most antibiotics prevent replication. They don't really kill live bacteria. They prevent them from replicating. So first of all, you have a challenge, so just with like a bacterial infection, of getting your treatment to the right place. Now with a staph infection, the bacteria is mostly at the surface, so administering the treatment is not incredibly challenging. Then you have an issue of trying to limit replication of the pathogen while giving your immune system an opportunity to kill it. And then another challenge you have on top of that is some of them, in addition to just feeding off the host, they're actually secreting toxins that just damage tissues and inhibit you from doing things. Now, in a antibiotic resistant bacteria, there's really the only thing we can do is try to encourage your immune system to fight it, because we have no really effective means by which to treat it. Yeah. Now, if you're talking about something like cancer, cancer is especially difficult because it's your own cells. Now they're different genetically, and they're a little bit different on the surface, so we're getting better at targeting those cells specifically versus your other cells. The problem is our most effective means of fighting cancer right now also kill your other cells. And so it's kind of a challenge. Like you can't just give somebody the dose necessary to kill all the cancer cells, because then you're gonna have enough what's the term I'm looking for? Not friendly fire, but you have enough collateral damage that it's just you can't do it. So that's a big challenge. On top of that, cancer cells replicate really fast, and they're immortal. I mean, they're expressing a lot of telomerase, and they'll just keep replicating time after time after time again. And every time they do, they become a little bit more mutant. So, yeah. Was there another question over there, Trinity or Lydia?
I have another question.
Oh, you had another question, Michelle, and then back to Emma.
So is carrot really good for your eyes?
Carrots? Yeah, so carrots have a vitamin in them that is good for eye health. But if you eat too much carrots, you concentrate that orange pigment and you turn orange.
Do you really? I eat so many carrots. Like
I eat carrots.
Eat some more and see how many you have to eat to actually turn orange.
I eat a whole bag of carrots.
Every day?
No, like a lot. Like every week. Okay. Nick, return one.
Maybe. Have you seen the episode of The Magic School Bus where
I freaking love Magic School Bus.
like you have to watch the original, not the Magic School Bus Rides Again? What's the nerdy kid that's always complaining about the Arnold? When Arnold turns orange because he's eating these like seaweed treats? Sorry, Emma.
Oh yeah. So like, what exactly is staph infection?
What is this? It's just it's the bacteria are eating your skin.
How do you get skin
Yeah,
cancer?
like I'm like super prone. I've gotten like 10 times.
Cancer? You've gotten
Yeah, so
Yeah,
it's
I got
it's a type of bacteria, Staph aureus, and it grows. And the way it grows is it's eating your skin, and then on top of that, your immune system is reacting to its presence, creating inflammation. And how do you get it? I mean, just being exposed to that bacteria.
Yeah,
Usually in an open wound, it needs to start.
Because I pull wingnails and that's what the doctor said I get them all the time.
Yep. So usually it starts with an open wound and then exposure to Staph aureus.
Oh yeah.
But Staph aureus, some of them are normal flora, like they're there all the time. They just, get a little crazy. Okay. So there are three main shapes for bacteria. There are rod-shaped. Rod-shaped bacteria, and we call these
This is red.
bacilli. Rod. My O is interesting, or what Emma's showing you is interesting.
Your eye is interesting. She showed me yeah,
Yeah. Except for rod's more like a cylinder,
Yeah,
but
it's the closest thing I know.
that's somewhere between a cone and a
Oh,
cylinder.
this is more of a rod.
No, it's not curved. Oh, yes.
Then we have spherical,
That's more like press.
and we call these
Solid rod.
coxy.
Solid rods. These are some solid rods right here.
Yeah, that's a great rod shape right there. That bacilli, yeah.
Is the last one like the silver?
Yes, spiral shaped. Yep. You got it.
I failed this test question.
Spiral shaped.
Is it the chemistry test?
No.
And this is spirally. Spiral shaped, spirally. And so these are the three main shapes. There are some bacteria that don't fit into one of these three categories, but these are the three main shapes of bacteria. And so staph is short for Staphylococcus. And so what shape do you think that is? Spherical. Coxy, Staphylococcus, or coccus. And then staphylo means several strung together, so you get a whole bunch of these individual bacteria, coxy, all strung together like a chain of beads.
It doesn't sound very good.
It doesn't, and it isn't. Even if you don't have an antibiotic resistant staph infection, they're still very dangerous because they spread really quickly, They grow really fast, and they're actively eating your flesh. Now, then you have Streptococcus, which is another genus of bacteria, and they like to eat skin as well, and they cause what's called the flesh-eating disease.
That's gross.
Yeah. It's necrotizing fasciitis. Necrotizing, which means death-causing.
That's what they got in Grey's Anatomy.
Emma and then Trinity.
Yeah. I have a question about leeches.
About leeches?
What category of animals do they fall into?
They're annelids, like earthworms.
Ew.
Okay. And like
They're in a different class. So inside of phylum Annelida, they're called, that phylum is called segmented worms. There are 3 classes. There's Polychaeta, and those are mostly marine, and they have like really long fibers. They look like little mustaches that move. And then, oh, thank you for that. And then you have Oligochaeta, which are mostly the earthworms and freshwater worms, and then you've got Heterogeneity, which are the leeches.
It's like burgers.
Not all of them are parasitic, by the way. A lot of them are predatory.
Leeches are gross. Or meat predatory.
That means they swim after other animals and they eat them. Yeah, Trinity. Okay, shh.
Oh, so skin cancer?
Yeah.
Does that, like, have something to do with like the flesh eating bacteria?
No, not usually. Not usually. It's just mutant cells growing out of control. Yes, Emma.
okay. Wait a second. Oh yeah, are moray eels like what are those? Like
They're fish.
they're fish?
They're fish,
They're
yeah.
eels, so
Okay.
they're in
They're bony fish.
the-
Osteichthyes. I don't know what family they're in, but it's class Osteichthyes. Yeah. And then they would also be They would also be in Actinopterygii, which is a subclass, and then they would be in Teleosteae, which is an infraclass. And then after that, I don't know. All right. Any questions about this? I need to erase this so I can project to you an image of these bacteria.
That'd be fun.
Because one thing that did come up in my evaluations when y'all were given an opportunity to write what could be improved about the course, several people wrote, it'd be nice if Dr. Engel were here more. So like if I needed help. I don't know. I just get the summary.
Did she say anything bad about it?
I put on the
What she said is that not every teacher's evaluations are like this.
Oh yeah.
Which means
So-
you have an amazing evaluation.
I don't know what all I know is what I have and with that statement. I don't know if that means other teachers are usually better.
No.
I don't know.
No, that means
I didn't think I wrote anything. I was just like,
really good.
my suggestion is to keep doing what you're doing.
Just keep rocking it, Dr. Engel. Okay?
Stay jazzed.
All right. Let's do this.
No. Ew.
Now let's see if it'll project on there. There it is. There it is. Okay. So here we have our spherical, our coccybacteria. And here it's actually showing you 2 of them stuck together. This is a Streptococcus, okay, where you've got 2 cocci associated together. Here's the bacillus, the rod-shaped. Oh, great example of this, Bacillus anthrax, the causative agent of anthrax, which you all aren't old enough to remember. Some of you weren't even born there, born then. But following 9/11, there was a lot of fear of additional terrorist activity, and people were getting envelopes, like people that worked in the government were getting envelopes with like white powder. There was a lot of fear that people were mailing anthrax around. Anyways. And then here's the spirally, the spiral-shaped bacteria. Okay?
Slide left.
I don't know.
Slide to the right.
I don't know.
It's just stretched out.
Yeah. Yeah.
What's the purpose of the first one, like merging? Is there a reason, or is it just like
The purpose, so bacteria oftentimes make what's called a biofilm. And so when they make a biofilm, they basically make a non-living material that surrounds the bacteria and keeps them safe and nourishes them. And in order to do that, they have to associate with other individuals. Yeah. Tori.
Okay, so you know that like
thing when some people get the little like worm type thing in their like skin or in their body or something? And
Or you're talking about a worm underneath the skin? Yeah, it's a
Yeah.
parasitic worm.
Yeah, is that kind of like that?
No, I mean this. So do you see these size? Yeah. Shh. So this width right here is 5 micrometers. A micrometer is 1/1,000,000th of a meter.
Oh, that's
So a micrometer is 1/1000th of a centimeter of a millimeter. And a millimeter, they're about 15 millimeters in the width of my pinky.
Like,
Okay, everyone look at my pinky. Everyone look. You see my pinky?
yeah.
You see it?
Yeah.
Can you see it?
Yeah.
It's about 15 millimeters.
Okay.
And so this is less than 1/1000th of one of the 15 millimeters that make up the tip of my pinky. Okay? These are really tiny.
Millimeters, you think it's-
So
These are
how
really
do
tiny.
those little worm things like fit in our body?
Oh, they come in as microscopic larvae, and they get injected underneath the skin, usually by mosquitoes or some other type of insect, and then they grow and mold and divide and become adults.
How long do you think it'll take from when you get bit
is
by
why
a
we don't
mosquito or
listen.
something?
A month?
until it-
A few weeks or a month.
This is why.
Every time you get bit.
Not if they're underneath the skin like that.
Okay.
So you're mixing two very different ideas. Oh, yeah. So yeah, if you've got a tapeworm, yeah, typically you'll poop out little segments of it. You won't poop out the whole tapeworm, but the segments. So tapeworms, this is kind of fun. The segments break off as a means of the tapeworm reproducing. So the segments get packed full of eggs, and then they break off, and then the eggs will get released somewhere, get an intermediate host, will grow to a point where then you eat the intermediate host and you get the tapeworm back. But the little pieces that break off are mobile. So usually when you find it in your feces or your dog's feces, it's because the little segments of tapeworm are moving. Oh yeah. If your dog, so it doesn't happen a lot here because there's not a lot of wild stuff for the dogs to eat here, maybe a ground squirrel or two. But if you live in an area where your dog will eat a lot of fish, like so I lived in Ohio for 4 years, and there are a lot of rivers and lakes, and the dogs will just get in there and grab fish. And so the dogs often get fish tapeworm, and then yeah, it's very obvious. Because you'll see, like if you saw a pile of your dog's feces from like 10 feet away, it would be moving. And because there are worms inside of it.
That's disgusting.
I mean, one way to look at it is it's gross. The other way is like, wow, this is really cool. I mean, you can't help it. It's a moving pile of poop.
That's disgusting.
Emma.
It's coming at
I
you.
was watching Keeping Up with the Kardashians, and Chloe's like, I want to get tapeworms like this way.
Yeah, I mean it's.
People do that.
So stupid.
Why do that?
Why does
That is the most Kardashian thing I've ever heard.
Why do people do that? I mean, it's
How do
It's
they
like that.
because they compete with you for food.
They
And so
compete with you for food, Tori.
Yeah, so they eat what's inside of your intestines before you.
I'll beat it for their intestines.
And so
I'll
if
win.
you can have beef tapeworm, and you can have a 20-foot long tapeworm, and it's hungry. I mean, a 20-foot worm, it's hungry. It eats a lot. Now, the thing you have to worry about if you're going to do the tapeworm diet is vitamin B12 deficiency, which will make you anemic.
Is a thing?
Yeah.
Tori, you literally just like put tapeworms in.
Yeah, that's
Well,
like
I mean, you take like a capsule, yeah, that has the larvae.
Why would you do that?
Isn't that dumb?
It's illegal in the United States,
Yeah.
but that doesn't mean you can't get it. Anyways, so if you're going to do that, you'd want to take a vitamin B12 complex so that you don't become anemic.
in the first place.
I can't.
That's really the only thing you worry about. So listen to this.
That's a wrong conversation.
Oh my gosh, we're not getting anywhere. If it grows,
It was their fault, by the way.
it can't because the ends of it are always breaking off.
And maybe
Right.
more.
So how do they
So you
get
never
Well-
get rid of garbage?
How do you get rid of it?
You can kill it. You take an anti-parasitic.
I'm not even 20.
It's very easy to kill a parasite that's in your
Addiction.
So there was a researcher that was doing work in Africa, and he wanted to infect himself with beef tapeworm
Why would you do that?
because he just wanted to. So he infected himself with beef tapeworm, and then when he got back to his lab in the UK, he swallowed a camera Like there are pill cameras that you can use that they'll do instead of like doing an endoscopy.
I would do
And it took pictures as it went down, and then you pass it, and then you can access the pictures that are on.
because you have to dig into your
And so it was really cool because he waited long enough. He waited long enough that the tapeworm had taken root, and you could see it, and you just got wonderful pictures of this tapeworm.
Can we see?
Yeah, I'll maybe show you it later.
No, I want to
Okay.
see some.
So another thing about bacteria, what they look like. Okay, so Emma, you asked, can we tell from looking at a bacterium whether it is beneficial or whether it's harmful? And this isn't like a hard and fast rule, but we've got two different type of bacteria. I don't know if you can see these worms. Now I got worms on the mic. Gram positive. Okay, right here, these are gram-positive bacteria, and the outermost layer is a cell wall. Here, let me turn the lights back on.
Can gram-positive be for any of the three types?
Yes, yep, any of the three shapes, you got it. Yep, okay. So these gram-positive bacteria, cell wall is the outermost structure. And where do you see this whole positive negative show up? You see it with grades, but then you see it with like medical tests, right?
Oh yes. Like
Right? So
swab for strep throat.
positive, negative, or blood type, swab for strep throat. And so what gram positive means is it means the gram stain took. The gram stain took. And so a cell wall is the outermost structure, and the reason for this is the gram stain targets. This is a fun word. You're gonna like this word, not targets. This one. Peptidoglycan. I like to go to Target in a red polo and khakis just to make people think I work there. And then I'm like, I don't work here. You pay me? And then they're like, oh, I'm so sorry, but really I went there dressed up like I did work
Wait,
there.
people actually come up to you like ask you stuff?
Oh yeah, all the time. I used to do it at Walmart, but then the people that would come up and talk to me, they were a little scary. So I don't do it at Walmart anymore. Because you can go to Walmart with like a blue polo and some khakis.
Yeah.
Yeah.
You do that
But
anyway.
Target and a red polo and khakis, it's fun.
Go to the Nike stores, we're all Nike.
Okay.
Shirt.
Yeah, but there aren't a lot of employees there. But you know how hard it is to find an employee when you're inside Target or Walmart and you're looking for something? Like, you gotta search for people. So they're like, oh, thank God. Do you know where this is? I'm like, nah, I have no idea.
You can just tell them someone. They're like, oh,
Yeah, it's like,
I
it's like I just keep pretending like I work there, but yeah, like I have no idea, I don't care. And then they're just like, man, I
No,
need to
you'd
go
be like,
tell
oh, I'll
somebody
take you
about
to
this
it. It's
terrible-
over here.
Yeah, I know.
Bro, I know work here.
Mark.
Like at one point, I have no idea how this happened because I did not dress in any way like this because I at one point someone thought I worked at Macy's.
Macy's?
Or Macy's, like I was.
I mean, that's a compliment. The people that work inside the Macy's store, they're usually looking pretty sharp.
But I wasn't wearing anything like they were. Like I was just like wandering around and like and then like someone asked me where such and such and like I don't work here.
Now, if you go into like Abercrombie and Fitch with some tight jeans and no shirt on, they'll probably think you work there too.
Oh, let's do a call.
Oh yeah, so is that definition just for gram positive? You were
Yes. Yeah. So well the gram. So the gram stain always targets peptidoglycan, and peptidoglycan is what bacteria cell walls are made of. So plant cell walls, y'all remember this from Honors Bio, what are they made of? From Honors Bio, what are plant cell walls made of?
no, that's wrong.
It's a glucose polymer, it starts with a C and rhymes with cellulose.
Cellulose.
There we go!
I was so confused. Wait,
Plant cell walls are made of cellulose. Fungal cell walls are made out of another material that starts with a C and rhymes with chitin. 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 You could call it chitin. It's chitin, but bacterial cell walls are made of peptidoglycan, which is it's also a carbohydrate. Chitin's a carbohydrate. Cellulose is a carbohydrate. Peptidoglycan is a carbohydrate. But this is specific to bacteria. This is the only place you find peptidoglycan in bacteria. And the gram stain targets peptidoglycan. So in gram-positive bacteria, the outermost layer is the cell wall, and that gram stain can actually access it because it's the outermost layer, right? Right. Okay. Right. Gram-negative bacteria. Check this out. These are gram-negative. And the cell wall's not the outermost layer. There's a membrane. Well, how do we want to say this? Membrane outside? Yeah. Membrane outside the cell wall. And you can see that illustrated here. Look at this. This is gram-positive. You see the outermost layer is that cell wall. Here's gram-negative. The outermost layer, there's a membrane outside of that.
Isn't that like plant cells?
Plant cells, if you have two neighboring plant cells, then yes, there's membrane on either side of the plant cell wall. On an individual plant cell, the outermost layer is the cell wall.
Okay.
Yeah, but you don't really find a single plant cell, right? They're all together, and so you've got membrane on either side of it. So you see this? Outermost layer, the cell wall. The gram stain targets the cell wall, so the stain will access its target here, and it'll be positive. Here, the membrane's the outermost structure. The stain cannot access the cell wall, so the stain won't take, and it's gram-negative. Okay? Almost all pathogenic bacteria are gram-negative. Almost all. Pathogenic, meaning disease-causing. Pathogenic bacteria are gram-negative. Now, this isn't saying all gram-negative bacteria are pathogenic, but almost all pathogenic bacteria are gram-negative. So if you have a gram-positive bacterium, there's a pretty good chance that it is not harmful, that it's good or it's neutral. And if you've got a gram-negative bacteria, there's a pretty decent chance that it's pathogenic. Okay, and notice there's one other structure outside of here. You see this? Here's our outer membrane. And you see this grass? That's a sticky layer. It's a sugary, sticky layer called the capsule, and it does a really good job keeping these bacteria adhered to your surface, on your skin or in your intestines, where they can just wreak havoc. You can just release toxins like crazy, eat up whatever they find. Okay?
I thought it was this person.
Does this make sense?
So foolish.
Okay. Any questions? Okay. There's one more structure I want to show you. But before I do, I need to erase everything that's on the board. Man, I think it's now this whole week we've had everybody in class.
Yeah. Whoa.
For an entire
It
week.
only took us about an hour late, but it works.
Bare minimum.
Thank you, everyone, for your engagement and your regular attendance to this course. That's a lot. All right, can I erase this? Hey, if I did a class as a 0 period, would you take it?
Yes.
Yeah. Okay.
I mean, it might conflict with morning workouts, so.
That's okay, keep doing it.
You're like, if I had an excuse to get out of morning workouts, let's do it. What if I did like a double 0 period that started at 5 in the morning? It's like a sunrise class.
To be honest, Dr. Engel, I probably wouldn't.
You probably wouldn't.
Probably wouldn't.
You know,
What if you did it in astronomy class?
Would that mean I don't have the first period? If I don't have the first period, I do it. If I still have the first period, then no.
I don't know. These are just some ideas that are roaming around. Okay, the last thing I want to point out.
-1 period.
Check this out. Do you see this bacteria and it's got all these fimbriae?
It's really pretty.
You see that? Fimbriae. Okay, those fibers. Some of these, so some bacteria do this, and when you see these are an indication that these bacteria are transferring genetic information. Fimbrii.
How do you spell that? Why can't I spell that?
Oh, it's FIMBRIAE.
Transfer.
Transferring?
I don't think
Is it just one R?
I think so.
I'm leaving 2. Bacteria are transferring genetic information.
That's the fiber.
Fimbrii.
Fimbrii. Yeah.
That's the fiber of bacteria.
Yes. Yep.
Okay, so it doesn't. If bacteria is not like transfer genetic info, and then
This is usually an indication that these bacteria are transferring genetic information. Because most of these Fimbrii are what we call sex pili. And so we call them sex pili because they're engaging in transferring genetic material. A bacterial version of sexual reproduction. Okay? And so that's the last thing as far as what bacteria look like to answer that question. And then I want to go back to why are they so successful? Why are they so successful? And somebody remind me, why are they so successful? They adapt quickly. And what are the 3 reasons they adapt quickly?
Rapid reproduction.
Rapid reproduction. Emma.
Genetic recombination.
Genetic recombination is common. And then Carrie. They're less resistant to mutations. Good. And so this MRSA, the methicillin-resistant Staph aureus, antibiotic resistance can spread very quickly through a population because those that don't die from the bacteria use have more offspring, right? But then we also know they can transfer that genetic material because genetic recombination is common. Yes?
Yes.
Okay, cool. Now I want to show you what were our 3 types of genetic recombination. We haven't talked about this in a while where we actually named them all.
Yeah, crossing over.
Well, crossing over is what we find in eukaryotes as they're making gametes. But our 3 types of recombination in bacteria. So remember, genetic recombination is common. That's one of the 3 things that makes bacteria adapt quickly. Yes? Okay. There are 3 types of genetic recombination in bacteria. And what is genetic recombination again? Well, crossing over is an example of it.
The mother and the father comes.
It's when you take 2 different chromosomes and you combine them into a single chromosome. Okay? So now you don't have to take the entire chromosome, it can just be pieces like what happens in eukaryotes during crossing over. But genetic recombination. 2 chromosomes, and I'm going to put in parentheses, or pieces. 2 chromosomes, or pieces, combine to form a single chromosome. That is genetic recombination. And there are 3 forms of it in bacteria. 3 forms of genetic recombination. One of which we saw in the Griffith experiment. Is that when it drinks in that genetic material, and that one's called transformation. And I'm putting this down here, leave some space. Do you see the space? Mimic what you see. There's some space because I'm going to put 2 above it.
I'm just gonna wait.
You're just going to wait.
Yeah.
You're going to get left behind.
No, it's okay.
You got left in the
I
dust.
don't know how much space.
Can we just make that one the first one?
No, because it's not the order of my slides. Actually, we could because I don't have a slide for this one. We totally could make it first, but I'm not going to. I made it third, you can make it first if you want. So transformation, this is DNA drinking, right? The bacteria take on DNA from their environment and incorporate it into their chromosome. DNA recombination. We saw this in the Griffith experiment and then Avery's follow-up to the Griffith experiment. That didn't work very well. I wanted to pull it like a sword out of a sheath and it stuck.
I'm sorry.
No, I'm sorry. You deserve better.
Yeah.
You deserve only the best, and yeah, you're not getting the best of me. Transduction. And in transduction, a virus injects DNA. In transduction, a virus injects DNA. And if that virus is, what kind of virus? It's going to, it's lysogenic. If that virus is lysogenic, it's going to lead to genetic recombination. That the genetic material from the virus will end up inside of the chromosome from the bacteria, right? Because lysogenic viruses incorporate their genome into the host genome. Lytic or lytic viruses blow the host apart. Okay, and the last one, anyone know? Anyone know? It doesn't even start with a T. You're like, oh, this is T, this is a T, that one should be a T. It's not, it's a C.
Just like asking, is it like a sexual?
It is. It's sexual. It's one bacterium transfers genetic material to another one. And look at this.
Conjugation. That makes sense.
Same root as conjugal.
Why would you put that?
Why would I put that in the middle?
Why would you squeeze that there?
Because there's plenty of space. I write huge and it fit just nicely. Yeah.
The tunnel thing that connects.
Yes.
I remember this.
Bacteria transfer DNA across the sex pilus. Sorry, I ran out of space. So in conjugation, bacteria transfer DNA across the sex pilus. So these are our three means of genetic recombination in bacteria. And we said that genetic recombination is common in bacteria, and it's one of the things that helps them adapt so quickly, getting different combinations. Different combinations. Not just any combination, but a recombination. Right?
Combination. Uncombination. Recombination.
Okay?
That's what I'm saying.
Questions? Thoughts? Do you love it?
Yeah,
You do love it.
we do.
Yeah, I know. Okay, so now I want to show you these. Yeah, Michelle.
How can they conjugate?
So one bacterium, I'll show you an image of this. One bacterium will transfer a small piece of DNA across the sex pilus. So it actually needs to inject the other bacterium with genetic material. And so you know now fimbrii? No. Fimbrii. Oh yeah, it's right there. So the fimbrii, basically they'll form almost like a needle that will go in and inject a neighboring bacterium. You see these things like sticking out? It'll go in and it'll puncture another bacterium, and then this one will inject it with some genetic material. And I'll show you that. Okay? Alright? Okay. They are hollow. Like the end of a scorpion tail. Oh, I mean, known as the telson, but you just call it the tail. And it's like a hypodermic needle. Okay, so look at this. Look at this. Look, everyone. Look, everyone. This is transduction. Transduction. Here's a virus, and it is injecting its genome. You see that? It's injecting its genome into the bacterial genome, and then it's in this one. It's lytic, and it's actually bursting this. But in this one right here, look, it's lysogenic. It's incorporating its genome. You see that purple and the green?
Oh, wow.
Is that hard to see?
Yeah.
All the way back there? Here, let's make this bigger.
What is the thing? Where does it go after it goes away? After it injects it? Does it like a
The virus? I mean, it's out of material.
and then it just dies?
Yeah. I mean, you could argue it was never even living in the first place.
It's like a bee. It stings you and dies.
Yeah.
I have a question.
Which seems like poetic justice, right? A bee stings you and it hurts real bad, but then you know it's gonna die.
I have a question.
Okay, now, shh. Now can you see it more clearly?
Little bit.
Yeah.
Here's transduction. The
Oh,
virus injecting its genetic material, and that genetic material is getting incorporated into the bacterial chromosome.
That's cool.
Genetic recombination. Now this chromosome is a recombinant of two separate chromosomes. Tori.
Why, I feel like, I know you've answered this before, but I don't remember, and it's off topic. Why do bees die when they sting?
Because when they sting you, their stinger has a barb on it that sticks inside your skin, and so when the bee goes to fly away, their stinger remains and it rips their body apart.
How long after they
No, there are species of bees that don't have barbs on the end, so they can sting you and then keep their.
like yellow jackets or
Yeah, but those are horny. Yeah, those don't have barbs, so they will keep theirs. But even some species of bees, Africanized honeybees usually don't have their stingers rip off. So they're hybrids of European and African honeybees. Issa and then Carrie.
Do they know they're gonna die when they
Oh yeah,
So it's suicide.
yes.
Worth it though.
But you could argue that the entire colony of bees is a single individual.
Whoa.
Yeah, Carrie.
So how would that
It's a Leviathan.
help them survive better? Like why haven't they?
How do they adapt that?
Yeah, why would
Because it's all the only thing that matters is the survival of the queen.
Cora taught me something.
So and bees, honeybees are haplodiploid, meaning the females are diploid, the males are haploid. And so sisters, all of the worker bees are females. Okay, I'm giving you guys good information and you're missing it. Having a conversation about March Madness. Okay.
It's just close.
Yeah.
So the all of the workers are female and they're very, very highly related. Very highly related because their dad, everyone's dad is their brother. So they're related. Their coefficient of relation instead of being 0.5, which would be normal for full siblings, is actually 0.75. So which means that the female bees, their offspring are less related to them than their sisters are. Right? Their sisters are more highly related to them than if they left the colony and went and found a male bee in another colony to reproduce with. Their sisters are more highly related to them than their own offspring would be. Unless they also mated with their male offspring. Okay? And so it's beneficial to them genetically. More of their markers would be passed off if another sister is born than if they have their own offspring. And so they'll protect the queen at all costs. And so the worker bees will give their lives to protect the colony and to protect the queen. Yeah, Issa.
So when you mean by highly related?
Highly related meaning greater than 0.5.
So like let's say you're 100% related. Is that
Well,
basically being the same?
Identical twins, yep.
Identical.
And even identical twins aren't exactly 100% related.
And so
you
to
gotta
be
wait,
more
brother,
related
sorry.
Is to look more alike, or to just have more of the same gene?
Both. Yeah. If you're more similar genetically, you're probably more similar morphologically
Like
as
a
well.
doppelganger, how does that work?
It varies. They could be very similar to you genetically, or usually it's just you ended up with a number of similar phenotypic features with different genetic programming. It's called genetic heterogeneity.
That's crazy.
Yeah, Tori.
So 2 questions. How long after a bee stings you does its body rip itself apart?
Oh, as soon as it tries to fly away.
Oh really?
Yeah.
And then another question is, do some bees, like I remember one time my brother was just sitting and a bee literally just came and landed on it and he was like, okay, like maybe it won't sting me, I'm not threatening it, and it just stung him after a little bit and it flew away.
Probably some chemical cue on his body frightened the bee.
He was like,
Like what would do that?
I don't know.
Place of glory.
Yep, I don't know. Okay, so that's transduction. Okay, the next one, conjugation. Oh, that one's okay. Can you see this one all right? So this is conjugation. This is sexual reproduction in bacteria. And so here you have this bacterium. Here they called it the mating bridge or the sex pilus. And so it's injected this neighboring bacteria with the sex pilus, and then it actually replicates this piece of DNA and injects the neighboring bacterium with it. Notice in order for this to happen, this has to have something on it. Can you see that? It's called the F plasmid. Do you see that?
No.
You don't see that?
No.
you see
It's-
what
Let me write it bigger. That right there is called the F plasmid. And a plasmid is just a small piece of genetic material. Here's the main chromosome. main chromosome. Here's the main chromosome, here's the F plasmid. This plasmid, in order for this bacterium to make a sex pilus and use it, this plasmid has to have what's called the F factor. Which the F stands, I think, for familial, and so it's talking about relationship and how the bacteria are going to move their genetic material. So let's say we have a methicillin-resistant Staph aureus. Okay? So this is methicillin-resistant Staph aureus. So this is a bacterium with antibiotic resistance. Okay? Does that make sense? MRSA resistance. Resistance? Does that work? Okay. Alright, so this is a individual bacterium that is resistant to that antibiotic. If it has that antibiotic resistance on an F plasmid, guess what it can do?
It can.
If the gene for antibiotic resistance is on an F plasmid, what can it do, Rick?
Like, not inject, but like go across the bridge.
No, absolutely. It can inject neighboring bacteria with that genetic material and make them antibiotic resistant through genetic recombination. And you're like, why on earth would it want to do that? And again, we talked about that these bacteria like to make biofilms, this non-living material that protects them, and they need multiple individuals to do that. And so they can pass the antibiotic resistance. Now they don't need to do it through conjugation. If a virus infects a antibiotic resistant form and is lytic and then goes and blows it up and takes some of that host DNA with it and goes and injects it somewhere else and decides it's going to become lysogenic, now that new bacterium can be antibiotic resistant. Of course the original is now dead because the virus blew it up, but you spread it. Or if a bacterium with antibiotic resistance dies and leaks its genetic material into the surrounding soup, non-antibiotic resistant forms can drink that DNA up through transformation. Okay? Does it make sense? Any questions? Yeah.
What about this?
Okay, well, any questions about this first?
No.
Okay. Let me pause the recording. You still working on that? Oh, just what's written. Oh, I never. All right. So I'm gonna block this for a moment. All right. Chapter 38.
17.
Chapter 38. It is a big jump.
Wait, 38 or 20?
27 to 38.
Is this our last chapter today?
It is.
What's it called?
This is our last chapter this week. This is our last chapter this month.
Whoa!
Chapter 38 is called
It's freezing.
Angiosperm Reproduction.
Oh my god.
Angiosperm Reproduction and Biotechnology.
That's kind of cool.
Aren't pythons angiosperms?
No, they're gymnosperms.
That's what you do. Angiosperm repro plus biotech.
So angiosperm reproduction and biotechnology. Now angiosperms are flowering plants. Angiosperms equal flowering plants. And what does it mean to be a flowering plant?
Fruit.
It means you produce flowers and you produce fruit. You produce flowers and you produce fruit. Emma.
How does like an apple come from a little flower?
I'll show you in a minute. Yeah, we'll talk about that.
That's the way it is.
It just does. Alright, so in chapter 38, we need to answer this question. What is a plant? What is a plant? And so here's what I want you to do. I want you to take a few minutes. I want you to work with those around you, and I want you to come up with a list of 10 things you know about plants. 10 things.
They're green.
Could be anything. Rick, with the people around you, write them down.
I was talking about people around.
10 things that you know about plants. In 2 minutes.
Thanks, right.
2 minutes, starting now.
Kyle, you're writing, right? Kyle, right. They're green. Photosynthesis.
You can't just write photosynthesis. That doesn't mean anything.
They utilize photosynthesis for. Write that. You actually can photosynthesize. They're living things.
Some are really tall, some are
They grow.
They need water.
They have roots that
They can die.
yeah, they can
and then there are like the flowers. Like the flower petals.
they can grow. There are many different
Wait a
types.
minute. I love how you just throw out random words.
Many different types.
Variation.
There are many different environments.
Water.
Life.
Big coal.
Alright. So what do we know about plants? What do we know about plants? Emma. They reproduce. They are living organisms, and living organisms reproduce. But plants, plant reproduction is a little unusual. Plant reproduction is a little unusual because we have what's called alternation of generations. Alternation of generations. And what this means is that we alternate between haploid state, which is 1N, and the diploid state, which is 2N. Like bees? Yeah, so female bees are 2N, are diploid. Male bees are 1N, are haploid. But plants alternate between these two phases. So plants will have a phase of their life cycle in which they are haploid, only have one copy of the genome. And they'll have a cycle of their life where they are diploid, having 2 copies of the genome. Now we, as animals, we are diploid. Now not all animals are, because male bees are haploid, but we are diploid. And the only haploid cells we produce are gametes. But plants tend to alternate their generations. They have a time as haploid and a time as diploid. Now, for most plants, the diploid stage is the dominant phase. Katie.
What is 1N and 2N?
The number of copies of the genome. So N is an entire genome. And so 1N means you have 1 copy. So for us, our genome is chromosomes 1 through 22 plus this X chromosome. Right?
Yeah.
So our genome, we have 1 copy of our genome that came from mom and 1 copy of our genome that came from dad. We are 2N because we have 2 full copies of the genome.
All in.
Actually, you only have 2 full copies of the genome if you're female, because you have 2 X chromosomes. If you're male, you actually have 1 full copy of the genome and then 1 partial copy with an extra weird chromosome. Yeah, Tori.
So if we do make a few haploid like cells or whatever, what do we do with them?
Use them for reproduction. So gametes are the only haploid cells that we make, and they're used to produce the next generation. Yeah. Okay, so haploid and diploid. The haploid stage of a plant is called the gametophyte. Gametophyte. And the diploid stage of a plant is called the sporophyte. Yep, you got it.
Like spores?
Like spores. Spore producing, gamete producing. Okay? The sporophyte is a spore producing structure, and the gametophyte is a gamete producing structure. It looked like that door was open. But I don't think it is.
It's just lesson.
If there was a shooter, where would we hide?
No.
Because there's like windows.
There.
We would die.
No, for windows.
Or we come out up on the roof.
Just behind
We'll
me.
find it. We can take
Yeah,
it.
Michelle.
Wait,
There's like one.
Guys, hey everyone, Michelle's asking a question and I can't hear her because you're so loud.
so what is gamete producing?
Yes.
What do you mean by that?
So the gametophyte makes gametes, male and female gametes, sperm and ova.
Oh, and that's
Sporophytes.
Oh,
Yeah, sporophytes produce spores, and you'll see this in a minute. Okay? So this is something we know about plants. And really, everything else we've dealt with in a different section. So I'm glad that this one came up first, because this is the only one we're going to write down. This is what chapter 28 is all about. Angiosperm reproduction and biotechnology.
Wow.
38, yeah, I said 28, I'm sorry. 38. Okay? So, shh. I heard several people yell out photosynthesis. I mean, it would be better to say plants photosynthesize, right? But that we dealt with in another chapter all on its own, and it'll come back. We'll deal some more with photosynthesis during our review phase. Plants, we talked about plant cell walls earlier today. Plant cell walls are made out of what? Cellulose. Mark.
They're in all sorts of different environments.
They sure are. Yep, they sure are. Okay. Shh. Now specifically angiosperms. So this is all we're going to do for what is a plant. I'm going to erase this. Why are angiosperms, and what's an angiosperm again? Flowering plant. Why are angiosperms so successful? So when you look at the number of plant species, the overwhelming majority of plant species are flowering plants. Okay? The overwhelming majority of plant species are flowering plants. And so the question is, why are they so successful?
I don't know.
Maybe.
Bees. Ooh.
Bees are a reason why they are so successful. Well, all plants are angiosperms. So angiosperms actually do a little bit better in dim light than other plants do.
They're probably just not as high maintenance as others.
So angiosperms do not require water for reproduction. They do not require water for reproduction. They also do not require wind for reproduction. Wind, yep, do not.
Like the movement of air.
Yes. So a lot of plants, have you ever been to a place where there are a lot of pine trees?
Yeah.
So when you're there in the spring or late spring, early summer, there's pine pollen everywhere.
That's like Sneeze Town right there.
Yes, because pine trees are not angiosperms, and they rely on wind to spread their pollen, which is the male gamete, to the female gamete. And so they just produce an enormous amount of pollen, do not require wind to reproduce. Because how do angiosperms reproduce, Emma? What did you say right before when I was like, why are angiosperms bees?
Not A, not C, but
B.
Angiosperms are so successful because animals spread the pollen. Animals spread the pollen. These are why these plants make flowers. The flowers attract animals because the flowers are full of nectar, material for the animals to eat, and when they come to get the nectar, they pick up the male gametes, the pollen, and they transfer it to another flower.
so what you're saying is the B movie is actually like accurate?
I mean, it's pretty ridiculous in how quickly things drop. But if bees stopped feeding on flowers, angiosperm populations would collapse. Absolutely.
That's what
Not that quickly though.
Not
Emma.
the whole jazz thing.
Yeah. So are all flowering plants angiosperms?
Yes.
Okay, so for like cacti that flower, are those angiosperms?
Same things. attracting animals. For most cacti, they're pollinated by bats. And so, so they're pollinated at night. When the bats come to feed on the nectar, they'll pick up the pollen and take it to a flower on another cactus.
Because like,
That's weird.
because
It is weird.
some cacti don't flower. Is that correct?
I mean, as far as I know, all of the cacti will flower at some point.
Oh.
Some of them don't flower every year because the conditions might not be right to actually take part in reproduction. And then some of them only flower for a matter of hours.
Oh.
So there's a cactus that produces a flower and they call it Queen of the Night. And it flowers for about 30 minutes.
How long? Like every night or?
Nope, just one night of the year,
Really?
usually, yeah. Trinity.
Why do bats feed on the cactus?
Well, because I mean, the same reason any animal would feed on it, because it's a good source of sugar and food, the nectar. Emma,
Oh yeah.
what's that? No. But some animals rely on that nectar. Yeah, Emma.
Okay. Why do some flowers close when it's dark? Is it just like-
Because the animals that pollinate them aren't active at night. So there's no reason to try to attract the animals that won't be there.
Go to sleep.
Yep. Okay, so why are angiosperms so successful? Animals spread the pollen, and animals spread what else?
Animals spread the seeds.
The seeds. You were gonna say that, Mark? I'm sorry.
Good job, Mark.
So angiosperms are successful because animals spread the pollen and animals spread the seeds. They don't rely on wind and water, meaning they can live and thrive in places that are dry and arid, and they can live and thrive in places that don't have enough wind to spread pollen effectively. And animals do a really good job of spreading that pollen. And animals do a really good job of spreading the seeds because angiosperms don't just make flowers, they also make fruit, and which are filled with the seeds. Okay? And the fruit is what attracts the animals because they want to eat it. And as they do, they take those seeds, and angiosperms have coated seeds, so the seeds survive the pass through the intestinal tract. And then the animal poops the seeds out somewhere else, and they can grow there and not compete with the adult. Okay? And this is why angiosperms are so successful. They do a great job of attracting animals to spread their pollen and attracting animals to spread the seeds. Okay? Does that make sense? Okay, so I want to show you this. We have a feature in angiosperms called double fertilization. Okay? And so, may I erase this?
Yes.
Okay. So much fun.
That was funny.
Isn't that what we were saying? This is so much fun.
They're mocking me because I said this is fun. So you first. Mocking me.
Oh, did I? I'm sorry. It was an accident. Okay. So,
That's funny.
all right. So here is an angiosperm flower. And there's no need for you to memorize the structures of a flower for this course. Now, maybe for other biology courses that you'll do in the future, you may need to memorize flower anatomy. What you need to be able to do is to look at this and to be able to interpret this. So here we have the life cycle of an angiosperm. You see? We've got the life cycle of an angiosperm. Within the flower, you have the male and the female gametophyte. And what is the gametophyte again? It's alternation of generations. It's the haploid life stage. And so the entire plant, you see this entire plant?
Yeah.
You can't see this from where you're sitting, but next to it, it says the mature sporophyte. And it's 2N, the normal diploid state of the plant. And that's what you see. That's the bulk of the plant is the sporophyte. Within the flower on that sporophyte are the male and female gametophytes, the haploid stage of the life cycle. And the male gametophyte, what do you think it makes? Male gametes. Because we said the gametophyte is a gamete producing stage, yes?
Yeah.
The male gametophyte makes male gametes. Here's the male gametophyte here. Okay, here's the male gametophyte right here. And so we've got, the male gametophyte is inside of here. All of these pollen grains. You see these pollen grains being released here? I know they're small. Yeah. They're the small little specks. They're small little specks to me. They're very small little specks to you. These are the male gametophytes, these pollen grains. And then inside of here, you've got the female gametophyte that makes the female gamete, the ovum. So within these pollen grains are the male gametophyte. Inside there, you have the male gamete, which is sperm. Yeah?
So it's just like the chromosomes that make them female and male, right?
In plants,
Like what
I don't know. I don't think plants don't have sex chromosomes because the male and the female gametophyte grow in the same flower. So the same plant individual is producing both male and female gametophytes
So
in
what
every
makes it
flower.
male and female?
The size of the gametes. The small gametes are male gametes, and so that makes that the male gametophyte. The large gametes are female gametes, and that makes that the female gametophyte. Okay? Now what's unusual, so this is very common for plants. Okay? Now for things that aren't flowering plants, they don't make flowers with the male and the female gametophyte inside. But if you look at a pine tree, so you were talking about pine trees, right? You have pine cones, and you actually have two different types of cones. Did you know that you have male cones and female cones? So pine trees have male cones with male gametophytes, the pollen grains, and they have female cones with female gametophytes. Same tree produces both male and female cones. So inside of here, so that's not unusual. It's not unusual for a plant, the main plant, the sporophyte, to produce the male and female gametophytes in the same plant. What's unusual for angiosperms is you get what's called double fertilization. So what does fertilization mean? We've talked about this term before with regards to gametes. When the sperm penetrates the egg, right? It fertilizes it. Do you remember talking about that before? So we start, here's the basic progression through generating male gametes. We get down here, we've got a male gametophyte, and we've got these male gametes inside of our pollen grains. Over here we've got our female gametes. Inside of here are eggs. You can see the egg right there. So we've got female gametes, we've got male gametes. And so typically you would have one sperm fertilize one ovum. Okay? And notice inside of this, here we have the ovum, the egg, and then we have all these polar cells. Do you remember when we were talking about meiosis in female cells? That they didn't distribute the cytoplasm evenly? And that's why the ovum is so big? Do you remember talking about that? No? Okay, well, we'll talk about that some more. But anyways, in angiosperms, what I need you to write down is this. You have what's called double fertilization in angiosperms. Double fertilization. And so in double fertilization, one sperm fertilizes the ovum. I abbreviated fertilize. Okay? One sperm fertilizes the ovum. A second sperm fertilizes a polar cell. So get that written down, and we will pick up here, not next week, because next week's spring break. Happy spring break, everyone. But we'll pick up on this on April 1st. April Fool's Day.
So you're gonna teach us all the wrong things. We're just kidding, none of that.
Maybe. I can turn this off because you don't need it for the double fertilization.
Just kidding.